Hello, welcome back to the Computational Genomics Lab. Today we are going to take our ATACseq peaks. We've so far made a function to upload our peaks and where they are, and how to find overlaps between all the samples in all 12 of our samples. So now we can take those peaks and find what genes they are associated with. So I'm using some more of our genomic ranges skills. In 09, we're going to go to lessons 09 and 03, peaks to genome features. And I'll open this up in my environment here. I did load some of these files because they take a minute, and I'll mention where that occurs, but you'll need to source the functions we made in the last two lessons. Okay, so I've already sourced that, and here's some more information on the peaks here. But again, this is how we'll start every time. Even though we have these handy functions, we still need to make the inputs for those functions, and that's the peak path, which we also make the FL. And you can import the peaks. I've already imported them, so they're here as my peaks. And we can see it's a list of GRanges. And then, so they would be loaded here with your import peaks function that you sourced in the R script above. So that should be all good, and then you can run your find common peaks. Looks like I have not done that yet, so I'll run that. It should take just a second. And now what that just did is find all the peaks that have overlaps in all the samples. That means they have to be the same in all the samples. There's examples of that in the 06 knitted document, 0906. And just some more information on the genome. This will be important if you're not in the class to get the gen code VM25. You can just Google gen code VM25 and you'll have the same annotation file as us. You want to be careful which annotation file you use for which genome. I've already imported that. This will take about 5 minutes, and that's denoted here. But it is the starts and stops of all the genes, the starts and stops of all the exons that we kind of went over briefly in our genome features classes before in 08. And so we're going to make some objects that will be really handy, like an object of all the genes. And to do that, we can go into our gen code grmouse, which is the entire genome annotation, and we can index out into the type column. Again, if you want to go back to 08, we went over some of these genome features, where they are. So it's in the type or at element data, list data in the gen code gr of genes. So we can now make just one GRanges of just where all the gene features are. And we can do that for just mRNAs as well by indexing into gene type, gene underscore type, where this was just type. And now what type of gene is it in the word protein coding matches, it'll make a GRanges of just protein coding genes. So let's do that. And the same for long non-coding RNAs, it'll find in that gene type the subset that are called link RNAs or long non-coding RNAs, which we can study in our Choose Your Own Adventure in level 10. Okay, so there's some results written out for you here, so you can help think about this a little more. And now the next thing we want to do is use the genomic ranges function called promoters, which is super handy. So what that's going to do is take those gene objects we just made, gen code gene, mRNA, link RNA, and all genes, and we can make a window of where the gene starts and 2,000 letters inside the gene and 2,000 bases upstream, and ask did any ATAC peaks fall in all the samples on that gene. So now we have a way of having an interval for the gene by giving it some flexibility up and downstream, and you can play with these windows. A commonly used one is 2,000 up, 2,000 down. It's kind of big. I would say 1,000 upstream, 500 downstream would be a little more conservative, but you can play with that. And that promoters function is just going to take our mRNA genes, GRanges, find where the start site is. It already knows where to look, and make a window 2,000 base pairs upstream and downstream of that start site. So let's do that really fast. It's amazing how much work was just done there that quick. Okay, so now let's say we want to find gene link RNAs that overlap with our common peaks. And so we can see that the length of our lincRNA gene promoters is 5,000, so we have about 5,000 to choose from. And we use our find overlaps function from genomic ranges and look at our promoter regions and find the overlaps between the common peaks, the ATAC peaks that occur in all 12 of our samples, irrespective of dox treatment. Okay, just to learn it first. Then we'll want to find out which ones are different in the next lesson. Okay, so first we're just going to find the overlaps of common ones and lincRNA promoters. And now if we look at that overlap, we see it's about 20%. So you already have a cool, and that's done here. Roughly 19% of the lincRNA promoters have an ATAC peak that are on. So those would be a fun group of genes to make a list of and ask in the RNA-seq data, are the TPMs higher for those genes that have an open promoter region versus those that don't, that might also be being expressed? Might be an interesting result. I actually don't know the answer to that result yet. I'm going to skip the looking at the at-2s, at-froms between the common peaks. I think I have the results written out here, but really it's just more overlaps of those gene annotation files that we turned into promoter windows. We can now use those to overlap with our ATAC peaks. That's the main lesson here. And so this is doing the same thing with gene promoters. Why not try it? Let's do the overlaps. So the length of the overlaps is 19,000, and basically almost, and there's a total of 54,000. So that's about 34% of all genes have an ATAC peak on their promoter, is what that's telling you. And again, this is all spelled out more simply in this text here if you want to read through it. Okay. So we can also do this with our find common peaks function, where I think what we just did is fine. It's only 2 files. We're comparing the genome annotation to a peak file. Not a big deal. But I do want to show you that if you had a bunch of annotations you wanted to compare it to, you'd make each one of those GRanges a GRange list, and then you do the comparison. And so that's what this is doing here, is we're going to make a list that is mRNA promoters and common peaks. So it's the same thing we did. Those are the 2 files we did find overlaps for. We're just loading them in as a list now. So now we'll have a GR list that has just 2 things in it, common peaks and mRNA promoters. Okay, so then now we can do the find common peaks function we made, and it'll find the overlaps in there. Okay, and let's check it. Roughly about the same answer. So 13,316 versus 13,700. When you make this many observations, it's okay to have, a couple hundred off here or there. And so here's just doing the other comparisons that are up above, comparing mRNA promoters to the common peaks, et cetera, et cetera. And here you just, every time you make a GR list, is all this is doing is saying sample 1, sample 2, and then we're going to find the overlaps of those 2 samples. And I think we're saving this one, so I'll run it. And so these are all the common peaks across all the genes. So anything that's called a gene and any ATAC peak that overlapped in that 2,000 upstream downstream window is now in that list, and we can see that is about 14,000 things there. So it gave us a few more. Again, that at-2, at-from, you got to be careful, and you could go back through that if it was, critical, which it is critical for your results. So here's what we'll do: is we'll print out a file that you can use in IGV to show where those overlaps are, and then you could put the bigwigs in for your peak files and visualize those peaks. And you can see more of that in 06 here. Okay, so let's start making bed files for all genes here that have a common overlap. It'll tell us where that peak was. Here we're doing common peaks with lincRNA promoters, finding the overlaps, find roughly about the same, and then we're going to export that as a bed file so we have where all those overlaps are. Now we're doing mRNA overlaps there, and we'll export those 12,223 intervals of where an attack peak overlapped in mRNA gene. Okay, so let's save all this stuff somewhere. I have a folder called results here. I saved all that. We'll use that to load in the next class, where we start trying to find peaks that are different between the 0 time point and the rest of the time points. So far, we've done everything that is common in all the samples, which is interesting in and of itself to say these are the open chromatin regions of a stem cell that are highly robust and reproducible across these 12 samples. Okay, so next time we'll try and find the different ones using more find overlaps. And I hope by now you can see you can either just do the find overlaps between two files, especially if it's a genome annotation file, or if you have a bunch of peak files, which we do, you might want to make a function to find the overlaps in all the samples, which is handy for almost anything I can think of when you have replicates to find the common peaks. So now we're going to try and find the different ones. All right, I'll see you in the next one.